cell signaling. It's a phenomenon in which information is relayed across the plasma membrane to the cell interior and often to the cell nucleus. It typically includes the recognition of the stimulus at the outer surface of the plasma membrane and then transfer of the signal across the plasma membrane and transmission of the signal to the cell interior, which triggers a response. This may include a change in gene expression, an alteration of the activity of the metabolic enzyme, or some kind of reconfiguration of the cytoskeletal, or a change in the ion permeability, or the activation of the DNA synthesis, or the death of the cell. It is actually a direct interaction of a cell with its neighbor or from the action of secreted signaling molecule. There are three modes of cell-cell signaling. One is endocrine signaling, paracrine signaling, and autocrine signaling. In endocrine signaling, the signaling molecules like hormones, they are secreted by specialized endocrine cells and carried through the circulation to act on the target cells which are present at distant body sites. For example, the different examples, the hormones which are secreted from the pituitary, thyroid, parathyroid, pancreas, adrenal glands, and gonads, they all are secreted, they carried through the circulatory system, and they act at a distant site to invoke the physiological response. The second type of signaling, mode of signaling, is paracrine signaling, in which a molecule is released by one cell and it acts on neighboring target cell. And the example for this, we can take the examples of the neurotransmitters. For example, it's a neuromuscular junction. The neurotransmitter is secreted by the presynaptic membrane of the neuron into the synaptic cleft, and then it acts or binds with the receptor present on the postsynaptic membrane and invokes a physiological response. The third type of signaling is the autocrine signaling, in which some of the cells, they respond to signaling molecules that they themselves produce. And a classic example is of the T lymphocytes, which respond to antigenic stimulation by synthesizing a growth factor that drives their own proliferation. Well, those were the three modes of signaling. And this signaling mechanism, they act through the receptors, and today I'll be talking about the G protein coupled receptors. They are also commonly called as the GPCRs. These GPCRs, they are so named because they interact with the G protein, and all the members of the GPCR superfamily, they have a specific characteristic that they have 7 transmembrane helical structure, and these 7, they're commonly called as 7TM receptors. These receptors, they have their amino terminal end towards the extracellular side and the C terminal end towards the intracellular side. There are thousands of different types of GPCRs which have been identified in the organisms. And these GPCRs, they have a diverse array, they can bind to diverse array of hormones, which can be both from the plants as well as from the animal system. These G protein coupled receptors, they are structurally and functionally related proteins. As I just discussed, that they are characterized by 7 membrane spanning alpha helices. The binding of the ligand to the extracellular domain of these receptors, they induce a conformational change on the cytosolic domain of the receptor, and this cytosolic domain then it interacts with or binds or associates with the G protein, which is present on the inner face of the plasma membrane. And the G protein then gets activated and it dissociates from the receptor to. The discovery of G protein actually came from the studies of hormones such as epinephrine, which regulates the synthesis of cyclic AMP in their target cells. And cyclic AMP is an important second messenger that mediates cellular responses to a variety of hormones. G proteins are heterotrimeric G proteins, which were discovered and characterized by Martin Rothbell and his colleagues at NIH and Alfred Gilman and his colleagues at the University of Virginia. These proteins are referred to as G proteins because they bind guanine nucleotide either in GDP form or GTP. These are heterotrimeric because all of them they consist of 3 different polypeptide subunits called as alpha, beta, and gamma subunit. And these G proteins, they are different from monomeric G proteins such as RAS proteins, which we'll be talking later. These G proteins are heterotrimeric G proteins, and they were discovered, purified by Martin Rothbell and his colleagues at the National Institute of Health and Alfred Gilman and colleagues at the University of Virginia. They are referred to as G proteins because they bind guanine nucleotide either in the GDP form or GTP, and they're described as heterotrimeric because they have 3 different types of subunits, that's alpha, beta, and gamma. The human genome, it encodes 21 different alpha subunits, 6 beta subunits, and 12 gamma subunits. And these different G proteins, they associate with different receptors. So this array of G proteins couple receptors to distinct intracellular targets. For example, the G proteins which are associated with the epinephrine receptors is called as GS because its alpha subunit, it stimulates the adenylate cyclase, and other G proteins, alpha and beta gamma subunit, they act instead to inhibit adenyl cyclase and to regulate the activities of the other target enzymes. The guanine nucleotide binding site is present on the G alpha subunit, and the replacement of GDP by GTP follows, that's following interaction with an activated GPCR, it results in a conformational change in the G alpha subunit. In its GTP bound conformation, the G alpha subunit has low affinity for the beta and gamma subunit. So on the exchange of GDP with GTP at the alpha subunit, it leads to dissociation from its complex. And this dissociated G alpha subunit, which has now GTP attached to it, that's also called as the activated G protein, and it moves through the membrane, diffuses through the membrane, and it activates an effector protein such as the adenylyl cyclase. In this case, the activation of the effector that leads to the production of the second messenger, that's cyclic AMP, by the enzyme adenylyl cyclase. And the other effectors that include phospholipases, those are the G protein is said to be on when its alpha subunit is bound to GTP. G alpha subunit can turn themselves off by hydrolysis of GTP to GDP and inorganic phosphate. This results in a conformational change causing a decrease in the affinity for the effector and an increase in the affinity for the beta gamma subunit. The hydrolysis of GTP in the G alpha subunit, this is carried out by the inherent GTPase activity of the alpha subunit. Well, we've seen that ligand binding results in receptor activation. Now, this activated receptors, they turn on G proteins and G proteins turn on effectors. Well, to prevent the overstimulation, receptors, they have to be blocked from continuing to activate G protein. So there is a system of desensitization. In desensitization process, it blocks the active receptors from turning on additional G proteins. And this takes place in 2 steps. In the first step, the cytoplasmic domain of the activated GPCR, it's phosphorylated by specific type of kinase called as the G protein coupled receptor kinase, and commonly called as GRK. GRKs from a small family, they form a small family of serine threonine protein kinases that specifically recognizes activated GPCRs. And the phosphorylation of the GPCR, it sets the stage for the second step, that's the binding of the protein called arrestin. Arrestin, it forms a small group of protein that binds to GPCRs and competes for binding with the heterotrimeric G protein. And as a result, arrestin binding, it prevents the further activation of additional G proteins.